that I can live them all one by one, one by one. Don't you go looking back, there's no looking back. And I don't mean to open every picture I saved up. I'm only trying to visit with you one by one, one by one. And I swear that I'm not gonna lose you now. I need you in my life somehow. And even when the lights go down, down, I could never lose you now. Even though we're not so close, yeah. Maybe you could pick me up still, 'cause I can't stop falling down. I can't stop falling down. Now I'm not gonna lose you. Oh, keep you in my eyes somehow. And even when the lights go down, down, I'm not gonna lose you now. I'm not gonna lose you now. I wish I could remember every second we had. I know I tried to count them all one by one, one by one. But you don't get them back, you don't get them back. Take every memory we've ever had. Oh, I wanna live them all one by one, one by one. And I swear that I'm not gonna lose you now. I'll keep you in my life somehow. And even when the lights go down, down, I could never lose you now. And even though we're not so close, yeah, maybe you could pick me up still, 'cause I can't stop falling down. I can't stop falling down. Now I'm not gonna lose you now. I can still remember all the lessons you gave me. I hope that I can live them all one by one, one by one. Don't you go looking back, there's no looking back. And I don't mean to open every picture I saved up. I'm only trying to visit with you one by one, one by one. And I swear that I'm not gonna lose you now. I need you in my life somehow. And even when the lights go down, down, I could never lose you now. could never lose you now. Even though we're not so close, yeah. Maybe you could pick me up still, 'cause I can't stop falling down. I can't stop falling down. Now I'm not gonna lose you. Oh, keep you in my eyes somehow. And even when the lights go down, down, I'm not gonna lose you, no. I'm not gonna lose you now.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. To the Honorable Professor Dr. Mimadi Martaniase, Medical Doctor, Master of Science, Clinical Microbiologist Consultant. A very warm welcome on behalf of the organizing committee to our distinguished guest speaker this morning, Professor Sokichi Matsumoto, Doctor of Dental Surgery PhD from Niigata University, Japan, and to all the participants today. Welcome to our guest lecture, Clinical Metagenomic in Infectious Disease Era, focused on Mycobacterium tuberculosis and non-tuberculous mycobacteria. Ladies and gentlemen, I'm Debbie Kusumaningrum. It is my pleasure to be Master of Ceremony for this event. We appreciate you taking time of your busy schedule to join us today. We hope the guest lecture will build up the knowledge about the next generation sequencing method for research mycobacteria. Then I will read the arrangement of the event. First, showing Indonesia Raya video. Second, showing Adelangga hymno video. Next, welcome speech from the chairman of the Department of Medical Microbiology, Faculty of Medicine, Universitas Adelangga. Guest lecture, and then question and answer, and the last is share photo and finish for the organization. Now the next item is singing the national anthem of the Republic of Indonesia and hymnal Airlangga. Committee, please. Now the hymnal Now for the hymnal Airlangga. Committee, please. Okay, ladies and gentlemen, the next item is welcoming remarks from represented by Dr. Pepi Dwi Endraswari. She is a Secretary Department of Medical Microbiology. To Dr. Pepi Dwi Endraswari, the time is yours.
Ya, thank you, Dr. Debbie. Assalamualaikum warahmatullahi wabarakatuh. Good morning, our distinguished guest, Profesor Sokichi Matsumoto, and selamat pagi dan salam sejahtera, Bapak, Ibu, Profesor, Dokter, and our guest lecture participants this morning. On this occasion, I represent Dr. Eko Budi Kundori as Head of Clinical Microbiology Department. He apologized for not being able to open this event. First, we would like to thank you to Professor Sokichi Matsumoto, who is willing to give a guest lecture this morning. And we know that topic Mycobacterium tuberculosis and non-tuberculous mycobacteria is very interesting because we know that Indonesia is a high burden country of TB, and now TB infection cases in our country are in the second rank after India. So we very often encounter patients with TB infections. And however, we know that currently there is still many limitations for the diagnosis of TB, especially for non-tuberculous mycobacteria infections. Currently, we often encounter cases with symptoms of TB, but the microbiological diagnosis for TB shows negative result. So it's possible that in such cases, the cause of NTM or other fastidious pathogens. So we hope that by studying this clinical metagenomic concepts this morning, it will be open up new insight for many research opportunities in the field. And in the future, we hope that it can even be used to diagnose TB or NTM infections and also other infectious pathogens. And secondly, this lecture is part of the adjunct professor of the Department Microbiology Faculty of Medicine, Universitas Airlangga, with the University of Niigata. We hope that these collaborations can continue so that it can provide benefits for both of institutions. And the last, we also thanks to Dr. Debbie and Prof. Nimade Mertaniasi who have initiated this event. Hopefully this event can be held smoothly and provide many benefits for all of us. Thank you, Dr. Debbie. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Pepi Dwi Endraswari. Now we continue to the main session. Keynote speaker, Professor Sokichi Matsumoto, DDS PhD from Niigata University, Japan. This session will hand over by our beloved moderator, Professor Nimade Mertaniasi, Clinical Microbiologist Consultant, as Chairperson of Clinical Microbiology Specialist Program. She graduated from Universitas Airlangga, and her research interest and activity focus on Mycobacterium tuberculosis, MOTT, and also antimicrobial resistance. And there are a lot of her international publication journal. All right, to Professor Nimade Mertaniasi, the floor is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi. Good morning, everybody. And the Honorable Professor Sokichi Matsumoto. And thanks to the committee, Dr. Debbie, Dr. Marsha, Dr. Mita, Dr. Suharyadi for this meeting. It is my great pleasure to be moderator in the webinar of guest lecture, Clinical Metagenomic in Infectious Disease Era, focus on Mycobacterium tuberculosis and non-tuberculous mycobacteria. The webinar is held in relation to a adjunct professor program collaborating between Faculty of Medicine, Universitas Airlangga, and Faculty of Medicine, Niigata University, Japan. We know Professor Sokichi Matsumoto, DDS, PhD, is one of our great partners since 2015 as adjunct professor. We really appreciate to your support, Prof, to our department, especially to our Faculty of Medicine Erlangga University in improving the education and sharing knowledge especially in microbiology. So hopefully the cooperation will establish and consist also in any situation like in this pandemic. Yes, and we hope all participants have idea to develop science and technology, especially in microbiology. Please start the curriculum vitae. Prof. Sokichi Matsumoto, DDS PhD, as Professor and Chairman of Department of Bacteriology, Department of Infectious Disease Control and International Medicine, Niigata University, Graduate School of Medical and Dental Science, and Department of Bacteriology, Niigata University School of Medicine. Education since 1986 till now in Niigata University School of Dentistry, and employment as a professor of bacteriology in Niigata University School of Medicine. Yes, so very great experience in microbiology. So we can guide to Professor Sokichi. Yeah, shall we start this lecture, Prof? Would you please, Professor Sokichi, to present your lecture? Please, Prof. Sokichi.
Okay, thank you. Introducing excellent introduction about the meeting and me, and I really appreciate for your organization of the meeting and the past collaborative activity with us. So that is actually fascinating. So we but anyway, yes, that is still ongoing, but that is actually nice activity. Yeah. Anyway, so and I thank you to the Dr. Devi, and she asked me about the lecture about metagenome analysis because we initiated collaboration with the metagenome. But okay, but so actually I have a deadline yesterday, so I didn't have a time to study about the metagenome. So in this lecture, I want to present our current progress used next generation sequencing. Yes. Anyway, I'll start the presentation, but can you hear me? And shall I? Can you see my slide and can you hear my voice well? Okay, let's start.
Yeah, Prof.
Thank you for introducing me. I'm Sokichi Matsumoto from in Niigata University now. And this is a picture when I visited the Airanga University. That was 2018, I thought. So Sudasono and Hatta statues and me. That was a nice visiting, nice experience for me. And I also visited maybe popular place, the big statue, and I'm here. By the way, I in Niigata City, and actually I presented this slide when I visited before. And Niigata City is located in the central region of Japan and opposite side to Tokyo. And we have a Shinano River here. That is the longest river in Japan, although it is not so long, but the longest river in Japan. Oh, this is in progress, but Niigata is written by this in kanji character, and ni means new, and gata means small lake, lagoon, pond. Okay. And we have many lagoons, small lakes here. And the point different from Indonesia is we have sometimes snow, right, in the winter season. And this is a Fukushima Lagoon photo presented in the public side of the Niigata city. But this is a picture actually I took by myself. That is a Fukushima Lagoon picture in the winter season. But in summer season, they become sunny. And this is lagoon. And in the spring, we have many flowers in the Fukushima Lagoon. This flower name is rape. And we can obtain the oils from these flowers. And swans come from Siberia, Russia, in the winter season. This is another lagoon or small lake in Niigata City. We call it Hyoko. And we also have some cultures. That is an old-time farmer's house in Niigata City. And the two women in this photo is actually come from Indonesia as a student exchange between us. And their name is, I think, Bale and Nanda-san. We ate lunch also in the old farmer's house refined restaurant. We also visited Nagaoka fireworks. That is one of the biggest fireworks in Japan around the Shinano River, and watched the fireworks there. The university located in such place, and our university was established long time before, more than 1 century. And this is a picture in 1914. And the building was ruined now, but the palm tree is still alive. Please check this. This is supported by the wood. That is why in one winter time, this palm tree was fall down by the weight of the snows. Actually, I picked this picture in that morning. That was actually Sunday. So because of the heavy snowing, I care about the laboratory and visited the university and found the palm trees were hauled down. But after that, this was supported and still living. At that time, so I have nothing to do in the laboratory, but I found the museum at that time and also found the bone that showed the tuberculosis carriers more than 2,000 years ago. That is in the museum in my university. I want to check their genome to see what's the ancient M tuberculosis. The laboratory was established also 1914, and now brand new, but not brand new, but so good. And the same age with Tokyo Station. And 2014 was 100-year anniversary. At this lab, as Nimade, Professor Nimade and coordinator show introduced. So we are focusing about the research on the mycobacteria, and we have a BSL2 lab and also BSL3 lab, and we can breed the animals there. We can check the drug efficacy and vaccine efficacy in this laboratory. That was picture before the corona era without mask. So as introduced, so TB is still serious problem, and around 1.4 million people die annually. And also NTM diseases are increasing. So our study is still important. So I show some data about the research of mycobacteria using NGS, next generation sequencing. So this is the some data. Maybe I pick it from some site. And the cost of the genome sequencing is reducing, but still expensive, but reduced substantially. And now we can determine the human genome in a day. Nowadays, we can make the library and sequence and assembly in the computer, and we can obtain the data. So, sorry, this is the Japanese, but NGS is a tool to determine the genome sequence. So the basic way is like that. So climbing on the mountain, like, so determining the long sequence is difficult and time consuming. However, fragmented DNA can be easily read and assembled in the computer, and we can obtain the data. That is NGS. Using this NGS, there are several methods. So one is simply whole genome sequencing. We can determine the human and bacteria genome in a day. Exome sequence, RNA-seq, that is transcription analysis. And now RNA-seq of the microRNA that become important because microRNA seems biomarker of the disease. And ChIP-seq is, for example, which part of the genome is covered or bound by DNA binding proteins such as histones. I don't know about this, but Haishi can estimate the structure of the genome. And this last one is related to the metagenome sequence. So 16S ribosome amplicon-seq. So we can analyze microbiota by amplification of the 16S ribosome RNA and identify the species of the bacteria in the particular sample or environments. So these are representative analytic way using NGS. So there are two types of sequencer now. One is short read, another is long read. So long read is feasible for the determination of the whole genome. However, short genome also useful because we can deeply sequence of the many fragments at the short time. It can be used for the, for example, RNA-seq analysis that can determine the transcriptional level of whole genes. So the representative of the short read sequencer is Illumina. And long read sequencer is we use PacBio sequencing. This can read around 10 to 50 kilobase pair of DNA, but its cost is high. Actually, we pay 1 million for one time sequence. Illumina is relatively cheaper comparing to these long read sequence, but still useful. So first time we used the next generation sequence is this paper. We determined the clinical isolate of the some NTM strains in 2012. The time before, we isolated several NTMs from the patients. So usually NTM diseases progress slowly. However, in several cases, we found rapid progression of the disease. And we isolate the strain, for example, 198, that also showed the hypervirulence in the mouse infection model. And we sequenced. This 198 NTM was actually M intracellular, and it contains around 5,000 of the genes and also plasmid. So we determined the genome, but at that time, we could not find which genes are responsible for the virulence of this pathogen. Just sequenced, but this should be the initial step for understanding of the pathogens. And actually this year, we also published another paper about the genome analysis of the NTM. So we extensively analyzed the M intracellular genome because, as I show, so we have reported the virulence of clinical M intracellular strains. In our experience, M intracellular strains are pathogenic than M avium strains. So, as I introduced, so mycobacteria have successfully adapted to human host environment as represented by the worldwide prevalence of tuberculosis and NTM. So M avium and M intracellular are called MAC because they show the similar characteristics and similar antibiotic sensitivities. Major and also important because they cause the pulmonary NTM diseases in Europe, US, and also including Asia. So identification of the genetic differences between species and strains provides basic information for investigating the cause of differential epidemiology, virulence, and pathogenesis. So as I show, and you may know, so recent advances in genome sequencing technology have enabled the comparison of the clinically isolated strain at the whole genome level. And this has revealed that some mycobacteria species have open pangenomic. That means the mycobacteria or most of the bacteria accept foreign genes to reconstruction of their genome. Human as well. So in Mark disease pathogen, so in avium, the genomic diversity is classified into several subspecies, including avium homonycis, paratuberculosis, and Shilbatium. So homonycis cause human diseases. The initially observed M. avium subspecies avium causes avian tuberculosis in birds and other domestic animals such as pig, cattle, blah, blah. But M. avium subspecies homonycis causes pulmonary infections to us. And M. avium paratuberculosis causes Jones diseases in the animals. So there is accumulation of the knowledge about the genome of M. avium. In contrast, other major etiologic agent of NTM diseases, M. intracellular, is still unknown. So M. intracellular, the comparative genome approach has not been reported for typing strain to date. Nevertheless, new relevant species and subspecies have been proposed, multi-locus sequence typing of single strain, for example, M. paraintracellare, Mycobacterium yongonese, and Mycobacterium indicus. So, however, the grouping of M. intracellular and its related strain is confusing, even in the NCBI genome database. As for February 2021, M. yongonese and MIP are registered as an independent subspecies of M. intracellular. For example, M. intracellular subspecies yongonese and M. intracellular subspecies intracellular. So they are still confusing. So we applied NGS analysis to classify to grouping of the intracellular clinical strains. We sequenced around 50 strains of the clinical isolates and also referred around 20 genome in the database. And this is a tree of the analysis of NGS data. And we found, we could not find M. yongonese in our libraries. Maybe M. yongonese is little in Japan. However, we found MP and MIP, but these intracellular can be classified in only two groups in our analysis: typical Mycobacteria intracellular and MIP group, that is M indicus or M para-intracellular. So they are proposed different species, but they seem same by our analysis. They also harbor the plasmid, but the plasmid presence of the plasmid is not limited to some group. They are distributed, many of the M intracellular strain. And this is a pan and core genome analysis of the sequence, and core genome level is stable and a little bit declined, but to M intracellular, these clinical strains obtained many foreign genes, and their genome diversity is still expanding. So they have pangenome. But again, importantly, we suggested M intracellular clinical strains can be only divided in two groups, typical and MIP group. So this is just grouping. So, but we also need to identify the function of each genes and want to identify the target of the drugs or vaccines. So we proceed transposon mutagenesis-based sequencing to know which genes are essential in M intracellular. So this paper was actually published in last year. So our associate professor, Yoshitaka Tateishi sensei, established transposon insertion to M intracellular. And this is hard process, but because need fine tuning of the infection. But he established this system and obtained many transposon randomly inserted M intracellular strain. And interestingly, this transposon only get in with single copy. So one gene is disrupted. But this transposon insert into the TA site. So TA site is frequent in the genome and also inserted basically randomly. So the genome is randomly one of genome of each bacteria is disrupted by the insertion of the transposon. And then we sequence using next generation sequencer, MiSeq, Illumina MiSeq, and identify the transposon located area, place. And for example, this is a theoretical insertion site And red one is a number of the insertion. And these genes were disrupted by transposon, but this is not. That means this gene is dispensable, but this is essential. So to make sure, we obtained 3 sets of the independent library. So this is a genome of the whole genome of the M intracellular of the type strain and the insertion number of the transposon. So transposon mostly inserted most of the genomic area, but some area cannot. And from these data, we identified around 500 genes are essential in M intracellular. At that time, there are 2 papers were published about the essentiality of the mycobacterial genome. One is M. tuberculosis, another is M. marina. And we compared the essentiality between M. tuberculosis and our M. intracellular and M. marinum and intracellular. And I'm not sure this is surprising or not, but essential genes are common. Some are common, but not all. So in M. tuberculosis, around 700 genes are essential, and only 280 were shared with M. intracellular. In marinum, around 300 genes, 304 were essential, and half are shared with M. intracellular. So essential genes are different in each among species, even in the mycobacterium. We also identified the essential gene of the clinical isolate. So that is a hypervirulent strain. And in this hypervirulent strain, the number of essential genes were lower comparing to the type strain. And interestingly, they are same M intracellular, but essential genes, half of the essential genes are different. So it suggests the difficulty of the drug development, but accumulation of the knowledge about the essentiality of the gene, we can focus of the genes that is commonly or definitively essential in the mycobacterium. And we checked these essential genes are related to the actual drugs for M. tuberculosis. For example, gyrase are target of the quinolone, and M genes are ethambutol target, isoniazid target, rifampicin target, RPOB. And interestingly, all of genes are also essential in M. intracellular. So target is good, but as you know, so M intracellular also tough, also tolerant to these drug treatment. So there are some other mechanisms why they become tolerant to these drugs. Target is still target. Recently, we also found another paper estimated the essential gene of Mycobacterium abscessus. That is also cause intractable disease and rapid progressor. So in their analysis, so similar to our analysis, some are essential genes are shared, but still species-specific essential gene exists. Okay. That is a transposon DNA sequencing analysis. I moved to the next result of us. So actually this paper was published this year, mainly conducted by the PhD student from Russia. His name is Alexander Irino. And he found the extracellular DNA of the mycobacteria. So why we started this experiment is we want to obtain the method determine the mycobacterial viability quickly because they are slow grower, relatively slow grower. So we found one paper that used the flow cytometry to analyze the viability of M. tuberculosis. So they used the flow cytometer and two dyes. One is cytoxgreen, another is CBAN. So cytoxgreen stains DNA. CBAM is calcium violet with an acetoxyl methyl ester that causes fluorescence when esterase is active. So esterase is a sign, esterase activity is a sign of the living of bacteria. So by using these dyes, they showed, so Mycobacterium tuberculosis was stained. If they are living, most of bacteria are stained only calcium violet. But if they die, if they die, the membrane will be broken. So the dye, cytoxgreen dye, can enter inside of M. tuberculosis and stain DNA. That's why they become positive. So that seems good method to estimate the viability of M. tuberculosis because FACS can be done in several hours. And we have a FACS machine in our laboratory. So we check that. But we cannot reproduce their data because, so this is M tuberculosis stained with calcium violet and cytoxgreen. This is dead M tuberculosis because they are treated with boiling, heated. So calcium violet staining was lost. Instead, most of bacteria become cytoxin positive. However, even living M tuberculosis, some of the calcium violet positive population become cytoxin positive. We cannot judge this is living or not. So we were confused and checked other mycobacteria such as BCZ, M intracellular, and M avium, and also MTB. They are all slow growers of mycobacteria. And we again found some living around 30 to 40% of living mycobacteria still positive to cytoxin. And we treated these bacteria with DNAs that degrade DNA, and the positive population, positive ratio was reduced. Not all, but substantially reduced. That means they have DNA outside of bacteria. That can be extracellular DNA. However, interestingly, this staining cannot stain any rapid growers we tested. So we could not see cytoxin positive, calcium violet positive population in these rapid growers. So that means extracellular DNA is specific for slow growers. That is interesting, but we don't know the reason why so far. But anyway, this is just a calculation of the extracellular DNA of BCZ, M intracellular, MTB, and M avium, and rapid growers, and their extracellular DNA level that was stained cytoxin was reduced by DNS treatment always. So this DNS treatment also reduced mycobacteria growth in case of BCZ, M intracellular, and M avium. We just cultured these bacteria on the media and added DNS or just control, and their growth rate is different. So maybe extracellular DNA participate in the enhancement of the growth. So maybe as you know, so extracellular DNA is known to be important for construction of the biofilm. So extracellular DNA is a component of the biofilm, for example, in the Pseudomonas aeruginosa. So biofilm resist antibiotics treatment. We checked DNS treatment also reduce the sensitivity. to antibiotics in mycobacteria. So result is not so remarkable, but in some antibiotic treatment, such as amikacin and isoniazid, DNA treatment enhanced drug efficacy. So they may contribute to the extracellular DNA, contribute to the drug tolerance of the mycobacteria. So we questioned what is the extracellular DNA of mycobacteria, and we used next generation sequencing at this point. So we purified the whole DNA and extracellular DNA from each slow grower of mycobacteria. So usually total DNA is obtained by melting the cell wall and membrane. However, we extracted extracellular DNA without disruption of the bacteria. So the size is a little bit smaller comparing to genomic DNA, but they still fractionated as a band in the electrophoresis and sequenced and compared with genomic DNA. So these are data. So in case of BCG, M intracellular, and M tuberculosis, so extracellular DNA and total DNA are same. However, interestingly, in M. avium, some extracellular DNAs, some genes are larger in extracellular DNA. So these suggest BCG, M intracellular, M tuberculosis, extracellular DNA may come from the dead bacteria, or we don't know, but our unknown system secrete whole DNA from bacteria. That was observed in the E. coli. But anyway, they are same. But M. avium, we found the difference between extracellular and genomic DNA, and we analyzed what these are. So this dot was located in this genomic region, and that was these, and these were PHI MAB1. So that means phage. So bacterial phage was released from M. avium and become a component of extracellular DNA. But anyway, we found extracellular DNA located specifically on the slow growers of mycobacteria. And the mechanism of the existence of these extracellular DNA is unknown, but one mechanism is in avium is releasing phage. And this eDNA contribute to the drug tolerance and forming mycobacterial biofilm. This is another data we published last year. We also used the NGS in this paper. This paper, we analyze the mechanism of deramide, a new TB drug, and found deramide formed a duct with NAD. So deramide is a new drug made by Otsuka Pharmaceutical Company. And this is a report published in 2012 in the New England Journal. It was approved for the use of the MDR or XDR treatment. This is the paper data, so comparing the placebo, addition of the deramide improved the conversion of the smear negative of the drug-resistant TB patients. So deramide treatment causes suppression of the synthesis of mycoric acid. That is true, but this phenomenon is occurred when the bacteria were treated with higher concentration of the deramide. So the MIC of the deramide is very low, so around 1 or 10 nanogram per ml. So in such condition, mycoric acid inhibition, synthesis of the mycoric acid, inhibition of the synthesis of mycoric acid is not occur. So we thought there is an unknown target of the deramide. So interestingly, this is a prodrug. That means deramide itself is not active. So after entering to MTB, it should be activated by this enzyme maybe. So this enzyme need cofactor cofactor, I don't know. F420 cofactor. Anyway, so activation of the deramide, for activation of deramide, many genes and cofactor are necessary. That's why it's a weak point of deramide, because the frequency of the drug resistance is high in deramide. It's much higher than isoniazide. That is a weak point of the deramide, because for the activation of the deramide, there are many genes, and many genes are no essential enzymes. They can be mutated. But anyway, but the deramide is still strong drug, and we chased the metabolisms after activation, basically mass spec analysis. And we resolved the puzzles of the mass data. And in this process, we noticed, so NAD and NADH level influence the efficacy of deramide and obtain the mutant that show the different MIC comparing to the wild type and sequenced by NGS, and identified one genes were mutated, and that was NDH related gene. And finally, we draw the picture of the activation of the deramide, and the deramide form the adduct with NAD. So NAD adduct deramide may inhibit the real target. So real target is still unknown. But anyway, this is the initial step to resolve the mechanism of the action of the deramide. But anyway, I want to strength is we use the NGS to find this phenomenon. This is a final chapter of my presentation. So this is a simple paper using next generation sequencing and estimated the genome mutation rates of M. tuberculosis. Actually, we published this paper last year. That showed higher genome mutation rate of Beijing lineage of M. tuberculosis during human infection. As you may know, MTB originated in Africa. It was estimated. So MTB spread out Africa over 20,000 years ago, following early human exodus from Africa. So this is only 7 arrows, but there are 7 lineage are known, and 2 are still in Africa. And 2 strain, 2 lineage are considered more pathogenic than others. One is lineage 2, spread in the Europe, and lineage 4, and lineage 2 spread in the Asia. I think Indonesia is a mixture of the Euro-African and Oceanian and Asian lineage 2. But anyway, so Beijing lineage is belonging to the lineage 2. As you know, they tend to easily become drug-resistant tuberculosis. So we obtained the clinical isolates of the Beijing lineage that was originated from 2 patients. But from these patients, this strain spread 1 to 3 patients. They rapidly progressed disease within a year. But other two progressed long time after from the infection, because this person only contacted with this person at this time point, not later time point. So this time span is 10 years. So we could conclude this strain latently infected this person for long time. And also after progression of the disease, person E, F were infected, and two or 3 years later, he also developed. This case is a reactivation of the same patient. So we sequenced total 1, 2, 3, 4, 5, 6, 7, 8 MTB strains and compared and found the mutation. This is a tree of phylogenetic, and actually B, C, D are near to A strain, and EF is a little bit and I forgot which strain, but one strain become isoniazide resistant after treatment of isoniazide. And that strain, we found large deletion of the CAT-G region. So maybe as you know, isoniazide is a prodrug like Delamide, and it must be activated by CAT-Z in MTB to express its activity. So in this strain, CAT-Z regions were largely deleted and become isoniazide resistant. So anyway, we compared rapid progressor that progressed tuberculosis within a year, and a slow progressor takes more than 2 years for disease progression, and compared the mutation rate and found, so mutation rate of the rapid progressor are higher than slow progressor. So maybe after, it's hard to conclude why this is happened. So one possibility is high mutation causes rapid progress of the disease, or they just multiply a lot. That's why duplication mediated mutation occurred. But anyway, the mutation rate is higher in the rapid progressor. And also interestingly, the mutation rates of these Beijing strains were higher than other M. tuberculosis lineage reported before. So we compared our data and previous report that estimated mutation rates of the lineage 4 in New England outbreak cases. So mutation rate is almost tenfold higher in Beijing lineage in both rapid and slow progressor. And we also checked the pattern of the mutation. And one of important finding is in the slow progressor. So we found frequent GC to TA mutation that occurred when the organisms are exposed to oxygen radicals. So it suggests during the latency, MTB bacilli is exposed to the oxygen radicals. So this is a conclusion. So anyway, we show the Beijing lineage has higher mutation ratio comparing to other lineage of M. tuberculosis. That may explain higher appearance rate of drug resistance of Beijing lineage. So I'm sorry I could not to study about the metagenome sequence this time, but anyway, NGS, maybe you know next generation sequence. Next generation means comparing to the Sanger sequence using gel electrophoresis. So this sequencing method is really powerful. And we actually used these NGS related data in most of studies we do. It can be used again whole genome, exome-seq, RNA-seq, RNA-seq of microRNA, chip-seq, high-seq, and metagenome. So that is very useful. Please try that to use. So, maybe 1 hour passed already. So unfortunately, this time I could not go to your place, but this is a picture when I visited you and gave some lecture like this. It's a nice picture actually. And also we have an exchange program, and this is a picture when my student, excellent student Amino-san, visited Airanga University and met Davis-san and Professor Nimade. And she have Toria-san got a job in Dupta University now. As she graduated Airanga University and then Professor Nimade's lab. And now she also got job in Japan and she is assistant professor in my laboratory. So exchanging. So we are happy to collaborate, continuous collaboration, continuous relationship. And please come to Niigata. And we have a snow. Maybe you don't have. Thank you for your listening and patience. That's all. If you want to ask something, they are welcome.
Thank you very much, Prof. Sokichi, for your excellent presentation. So first, I resume your presentation, Prof, before question from the participant. Yeah. So firstly, this is your excellent presentation in about your nice city and your culture in Japan. So very nice for us, Prof. Yeah. Yeah. And the other one is your Niigata University School Medicine is the big history from your university. And the second one is important one for us is your transfer of the big knowledge from you about the big role of new generation sequence and many methods in new generation sequence. So the others method or equipment is Illumina, MySec, etc. So the role in whole genome sequence, RNA sequence, transposon, and microRNA or 16S-RNA amplicon sequence. So the big role of the new generation sequence. So one is the role is the differentiation for the strain, species, subspecies, and diversity, and the mutation. So could be differentiates the virulence or virulent strain, basic for pathogenesis for TB, and the big role in the epidemiology, yeah, Prof. Epidemiology. So like the phylogenetic tree role in the epidemiologic role, and many you mentioned about the function of the protein and of the especially on the essential genes for metabolism and the role of the target of anti-TB drugs. Yeah, and you mentioned the new for us information. New for us is the flow cytometry method for antibiotic response for rapidly assess sensitivity to anti-TB drug. And the other important one is the role of extracellular DNA, yeah, bro. Extracellular DNA. We study from the new generation sequence. So it's the new for us, extracellular DNA. So some different in rapid growers and slow grower. Yeah, extracellular DNA could be a specific marker for rapid growers and others, and the growth and the role of in the growth for the extracellular DNA and the role in biofilm. It's very excellent, bro. And from the NGS, whole genome or the fragmented is important for the lineage related strain, and could study about the epidemiologic relationship. And the other important one is for the treasure for the spreading and for the spreading of virulence, strain, and drug resistance, all the road and clinical many benefits here. Thank you, Prof, for the excellent presentation. And we hope the participant could be questions to Professor Sukichi. Please shake hand or please active in the question. Yeah, would you please the question for Professor Sukichi? Ya, ya silahkan mungkin partisipan.
Good morning, Prof. Can I ask
Oh,
to Prof. Sokichi?
yes, please.
Okay, thank you. First of all, it's an honor for me to be here today. Thank you for Prof. Nimade and Prof. Sokichi for very well presentations. My name is Frendi and I'm master's student in tropical medicines at Airlangga University. And allow me to ask my questions. Okay, so first, Prof said that there is a possible for the extra chromosomal genomes to play a role in the virulence of MTB. And based on the previous article that I read, it states that the virulence of MTB is caused by a direct mutations that is result from the antimicrobial treatment. So a direct mutation happens to the chromosomal DNA of the MTB directly. And there is still no proven that the mutations in the plasmid can be directly transferred to the other microbes. And in this presentation, I just found that the extra chromosomal DNA, such as plasmid, can play a role in the virulence of MTB, which is a very great and significance of discovery, Prof. And so my question is there any possibility that the extra chromosomal DNA of MTB can be transmitted to the other DNA, to the other bacteria, so that it can cause transmissions of resistance to the other bacteria? Okay, and probably I need more information regarding the mechanisms. Well, that's all for me, Prof. Thank you so much for your presentation.
That's a very interesting idea. I didn't notice, but yeah, that there is a possibility. So, for example, we found M avium and M intracellular has extracellular DNA, and their genome is very rearranging. So they have a large pan genome, and their genome capacity is very plastic. So they can accept foreign DNA, and also, as you suggested, extracellular DNA can be captured or enter to the other bacteria especially in the environment. And as for M tuberculosis, so we know that there are few genes transferred to M tuberculosis from foreign organisms. So there are few. There are still small foreign DNA in the MTB genome, but the level is very less compared to other organisms. That is, I consider the reason is because the habitat place, their living place is in our lung. So lung, in the lung, there is no other microbes, only our genomes. So they have a chance to obtain other genome except for our DNA. And maybe so MTB in the lung or in our body has little chance to give their extracellular DNA to other organisms. But that is a theory, so I think there is a possibility because there can be our unknown communications. Yeah, but your idea consideration is very interesting. Thank you.
Thank you, Prof. Very well understood. And Prof. Mati, can I ask another questions?
Yeah, please.
Okay, thank you. Okay, Prof, regarding the new methods for detecting the resistance by using cytometer or cytoflow cytometer. My question is, what is the affinity for this method compared to the previous method? Well, we used to perform the DST or drug susceptibility test by using a culture or a PCR test. And I just found that this method can actually be used to identify the resistance microbes. All right, so what is the affinity? Is it better than the previous method? Thank you, Bro.
That's a good question. Actually, so PCR detects DNA. So DNA exists even after bacteria death. So PCR test basically cannot distinguish live or dead, living or dead bacteria. So, and PCR test also, for example, in the gene expert, that can be based on the PCR, and it can distinguish the rifampicillin resistant strain and sensitive strain. But it cannot determine the isoniazide resistant strains because more than 95% of the rifampicillin resistant strain contains a mutation in the LupoB gene. However, isoniazide resistant strains, some are, as I showed, so some had a mutation in the CAT-Z, but some had a different mutation in other genes. So it is difficult to check all of resistance by PCR-based method. So actually PCR is quite powerful technique. So, but it is not perfect. So basically the best method is culturing. We can actually see their sensitivity to drug or resistant to drug by culturing. But the problem is time consuming in case of mycobacteria. So this FACS method is complicated actually, but the merit is, as I show, so it can be done in a day and using only very small bacteria that is only 0.0005 OD. We need very tiny amount of bacteria. And merit is we can distinguish, say, are living or not by using calcium violet. But calcium violet esterase activity judged by calcium violet also remains after bacterial death. That's one of weak point of this method. But basically, that method is interesting and rapid, and we can see the character of the bacteria by the different way, different scope from culturing method and PCR. So checking the bacterial viability or resistance by the different method is informative and interesting. Maybe we can see, maybe we can get another finding. And as you know, so the mass spec also very powerful tool now. Mass spec analysis can distinguish the bacterial strains in a short time. Sorry for long response, but it's a very nice question. Thank you.
All right, Prof. It's okay. I understand right now. Thank you, Prof.
Thank you, Miss Brandy. Your good question. Ya, mungkin sambil menunggu any question, ya, itu memang mungkin bisa ditambahkan ya, in Indonesia. Sorry. Jadi begitu ya. Jadi memang perlu diteliti lebih lanjut ya, Saudara Brandy, Mr. Brandy ya, about the extracellular DNA, about the transmission of virulence like other, because could be study in vivo ya, in vivo in the pulmonary tissue, could be the different mechanism or some different mechanisms that Prof. Sokichi focus that usually less transmission about the genes of Mycobacteria. Yeah, Saudara Ferendi, Mr. Ferendi, and about the flow cytometry, yeah, could be some of method, and the viability is important for the antibiotic sensitivity test. So we compare about the molecular and viability like culture methods. Yes, thank you, Prof. Sukichi, and the others. Yeah, question.
Baik, terima kasih, Prof.
Thank you. Yeah, other question from Dr. Rahmi. Yeah, other question from Dr. Rahmi, PhD student.
Yeah, using SC6 RNA and TB infection from clinical license. Yeah, I think so.
RNA.
Interesting.
RNA.
Sensei, this is difficult to doing. This is they maybe have a lot of money to doing sequence analysis, special for TB infections. Is
Yeah,
possible?
but as I presented, so the price is down now. It's still expensive, but it is available. And but your proposal is maybe many person thing. So someone already studied the analysis. However, so the data is not always same, and that they are depending on the sample and area and person who proceeds. So it is, I think it is still variable to do it. But that is a very important trial, and especially in the field of the cancer, many people find out to the biomarker of the cancer for the diagnosis, for early diagnosis. So yeah, it's an important trial. Huh? Yeah.
Is this possible for applying in Indonesia?
Yeah, I think so. Yeah. If Dr. Davy is now preparing the sputum sample for the NGS for our collaboration, but you can prepare the sample. Maybe we can support your analysis. I'm not sure that you have a NGS machine or not, but if you have, you can do it in Airanga University. Or if you send the sample, we can order the company to proceed it. So it can be done.
Thank you very much, Sensei.
Thank you, Dr. Rahmi. Yeah, Dr. Rahmi is our alumni from Erlangga University, but right now get the study program on PhD. Yeah, thank you, Dr. Rahmi. Yeah. Any question, please?
Excuse me, Professor Made, can I have a question directly?
Oh yes, thank you, Dr. Lydia, please. Yeah.
Okay. Thank you, Matsumoto Sensei, for the nice presentation. I'm interested in your explanation about flow cytometry method to predict the resistance to antimicrobacterial tuberculosis drugs. So, if I understand correctly, the purpose is to detect the resistance to the drugs much more rapidly compared to the culture method. Am I correct?
Yes, originally, yes.
Yeah. So if so, it means that the Mycobacterium tuberculosis will be exposed for some times before, will be exposed to the drugs before flow cytometry analysis, right? So
Yes.
how long do we need to expose this bacteria to the drug? So the difference of the times will be how can I say, it will be very useful for us if it's so. Thank you.
So as maybe we consider the same thing as you considered now. So we want to know the viability after exposing antibiotics. So we checked, but unfortunately, we have not succeeded in the rapid assessment yet, because, so as I show, so that after heat killing, bacteria completely lose the calcium biol to staining. So esterase is inactivated soon by heating. However, for example, esterase is still active after treatment with isoniazide and rifampicin. So takes several days to completely lose their activity. So it is still time consuming. So we need to brush up the way to find the viability of MTB by FACS method. So maybe calcium violet is not enough for rapid estimation of the drug-killed M. tuberculosis. So we need to find out other dyes to stain. Please try that. Please check other dyes. There are many indicators in this world. So that's also important. That is interesting. So this is not established method. Is that okay?
Yes, thank you so much. Actually, I have another one question. I'm sorry if I don't understand correctly, but in your very first presentation, you mentioned about the some genes are essential for Mycobacterium tuberculosis or intercellular, and the other are not essential. So actually, what is the method that you use to define that these are essential genes or not? Are you using knockout system or how?
Yes, yes. We randomly knock out the genes. So we use the transposons that insert randomly to the mycobacterial genome. Because that is inserted randomly, it only recognizes TA, chimine adenine sequence. So chimine adenine sequence is very abundant in the genome in any of organisms, including mycobacteria. So we collected more than 20,000 isolates. So each isolate has a different disrupted mutant. And gathering around 20,000 mutants, then extract the DNA and sequence by NGS. And we can identify the transposon where the transposon was inserted. So transposon insertion means the gene was disrupted. That means the gene is not essential. So, but the essential gene, there is no insertion of the transposon. So we can determine which genes are essential or not by transposon randomly mutation and combination with NGS analysis. Does that make sense?
Yes, thank you so much.
Makasih. Thank you, Dr. Lydia. It's also our alumni from Erlangga University, Prof. Sokiji. Yeah, so the good question for Lydia. Yeah, sorry, Prof. The other from the Dr. Daniel from Catholic University Atma Jaya, Indonesia. Yeah, please, Dr. Daniel, your question about, is there any specific universal extracellular gene one that can be packed in diagnostic package and be used in clinical diagnostic? Yeah, Prof.
It can be, for example, I noticed now is combination with PCR. So extraction of the extracellular DNA and PCR, it can be used for clinical diagnosis. Maybe you know, so the only heating sample can be used the template of the PCR. I thought such easy method can be useful is maybe the pathogen has extracellular DNA. It's just thought, but well, anyway, I think that kind of nice idea. Because DNA is a huge source of the diagnosis, and it is exposed on the bacterial surface. It can be used for the diagnosis, yes. Thank you for your question.
You're welcome, Prof.
Ya, thank you Dr. Daniel. Jadi mungkin kita harus meneliti lanjut ya. Tadi Dr. Lydia juga tertarik ya, interest for extracellular DNA, Prof. And about the role in diagnostic and others. Yeah, could be some study for this role, yeah. Yeah, sorry, Prof. From the other one is Mr. Wahyu Herlambang. Is
yeah, I lead
program yeah in Erlangga University too. Yeah, Dr. Daniel is our alumni too. Yeah, yeah, please, Prof.
Yeah.
Wahyu.
So in this presentation, I just talked about the essentiality of the genes and intracellular when they are cultured in vitro. However, we can use TNSEC analysis for identification of the virulence or drug resistance. For example, after obtaining the transposon mutant library, one half is cultured in vitro and half is injected to the mouse. And we can compare the transposon mutant library in the culture and in mice. And maybe we can find out some genes are lost in the mice. So that is a virulent gene, at least in mice, maybe involved in the pathogenesis of the human as well. But anyway, so similarly, we can identify the we can identify the drug resistance related thing using transposon mutagenesis library. So it is really powerful tool to identify the virulence and drug sensitivity. We can change the condition, environment, mouse, in vivo, intracellular, or drug exposed condition, and then analyze it by NGS. Is that okay?
Okay, Prof. Thank you for your answer.
Ya, ya, Saudara Wahyu, Mr. Wahyu, ya banyak yang dijelaskan Prof. Sokiji ya. Jadi banyak sekali nanti peran-peran transposon dan sebagainya ya, belajar dari NGS ya, NGS role, mutation, transposon, dan ini ya, etc. ya. Ya, ya, thank you.
Thank you, Prof.
Saudara Wahyu, ya, thank you. And yeah, yeah, please, the other question. Any question? Yeah, so some of chat, Prof, from Dr. Daniel. Yeah, need any collaboration with many university in Indonesia, Prof. Yeah.
Oh, thank you.
Dr. Lydia still in Kobe University, Prof. Dr. Lydia is our alumni. Ya, Dr. Lydia masih di Kobe University ya. Ya.
Masih, Prof.
Ya, Dr. Lydia still in Kobe University. When you
University.
PhD?
Already finished, Prof. Thank you.
Congratulations for Dr. Lydia. Yeah. So it's in the post doctoral program.
Yes, now I am in postdoctoral program.
Nice program. Yeah. Any question, please? Yeah, because Dr. Debbie, is it the time is?
Is it okay, Prof? Yes.
Yeah. Yes. So because the time, Prof. Sokiji, because the time, so we will finish this lecture. So we hopefully that this kind webinar lecturer could be continued, Prof. So for the effort to our student and our alumni to develop science and technology, especially in medical microbiology. So once again, I say thank you very much to all of the participants. So right now about the 161 participants from our collegium, from our student. So thank you very much once again, and thank you for your kindness in this lecture and many experience and transfer of your knowledge to our student. Yeah, so thank you for all participants for your attentions. Yeah, so I will back the time to Dr. Debbie. Yeah, please, Dr. Debbie, for closing this lecture.
Okay, thank you very much, Professor Nimade Mertaniasih. Alhamdulillah, all the case session of sketch lecture has finished. Now it's time for take a photo together. For our participant, please turn on your camera to be documented. Well, there's a 7 layer, I think. Okay, I'll call.
Senior.
Yeah, please turn on your camera.
Yati from Universitas Pajajaran, Prof. Sokiji. Dr. Titi, terima kasih.
Good morning.
Terima kasih. Terima kasih.
Okay, I'll come. One layer. Please prepare. Smile.
A camera. Wow.
Yes, please, bro. 1, 2, 3, 4, 5, 6. Okay, now. Yeah.
Wow. Please send a picture.
Oh, yes, of course.
Thank
And
you
to
for
all,
inviting and hearing.
See you. See you.
See
and to
you.
all participants, please don't forget to fill out the Google Form absence, and the certificate will be sent to your email. All right, ladies and gentlemen, as the saying goes, everything that begins has to end. May I thank you for to all the speaker, to the moderator, for your active participation in this session. Thank you all for coming. I hope you all enjoy the program. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you.
Thank you, Prof. Sokiji. Thank you.
Thank you, Prof. And thank you, Doctor.
Participant. Thank you, Miss.
You coming?
Thank you, Prof.
See you soon. See you again.
See you.
Bye.
Bye. Ya, Dek Kurnia.
Iya, Prof. Gimana kabarnya, Prof? Semoga sehat selalu.
Nanti kolaborasi juga ya.
Amin, amin, Prof.
Gimana kabarnya di Malang?
Hujan, Prof. Gerimis.
Ya.
Sehat-sehat ya, Prof.
Ya, semua sehat ya.
Iya, alhamdulillah.
Juga ya.
Ya, Prof. Terima kasih banyak doanya, Prof.
Terima kasih, Dokter Gita. Terima kasih, Dokter Marsha. Terima kasih, Dokter Dedi. Terima kasih, Dokter Suharyadi. Sukses ya. Ini ya, jalannya mudah-mudahan nanti ke depan bisa ada guest lecture lagi ya, Dokter Dedi ya. Sambil Dokter Dedi sekolah. Nah, itu udah ditunggu tadi ya. Nanti mungkin Dokter Nita kalau sekolah ke Niigata, tadi bagus sekali ya kampusnya. Kebetulan saya sudah pernah ke sana. Ya, Dokter Marsha juga ya, mungkin ya. Dokter Pepi, Dokter Debbie tentunya. Mudah-mudahan sudah tidak lockdown ya. Tapi kayaknya ke Jepang kalau untuk student itu waktu lockdown pun ada dari kedutaan diizinkan.